Hi students, we're going to take a look at the Kelvin cycle next. We've just finished the light reactions, so I'm going to move to our PowerPoint presentation and we'll focus on the Kelvin cycle. Now the Kelvin cycle is named after the person who figured out the process. This guy's name is actually Melvin Kelvin. Now, I'm not sure whose parent would pick the name Melvin Kelvin, but you know, it makes for a fun story now, right? So the Kelvin cycle is what follows the light reactions. Remember, in the light reactions, light energy was used to make ATP and NADPH. Water, it was split in the process. Now, the Kelvin cycle occurs out in the stroma. So we're still in a chloroplast, but we're in that fluid matrix, you know, that fluid-like material outside of the thylakoids. During this process, carbon dioxide is, and we're just going to use the word fixed here. Carbon dioxide is grabbed from the environment and put onto an organic molecule. We say carbon dioxide is fixed. During this process, NADPH that was made in the light reactions. NADPH is like a shuttler of high energy electrons. It is going to be able to donate electrons in a redox reaction. NADPH is oxidized. That means it loses electrons. It's oxidized to NADP+. It loses electrons, while this other molecule, 1,3-bisphosphoglycerate, is reduced to something called glyceraldehyde 3-phosphate, G3P. This is a sugar. Hey, in this redox reaction, NADPH donates electrons to make a sugar. We have a redox reaction where we've got a molecule being reduced to finally make a 3-carbon sugar. We do it. Yes, that's photosynthesis right there, puppy. And ATP from the light reactions, whoops, sorry about that, guys. ATP from the light reactions is used as an energy source. Oh, this whole process is going to take some energy. No surprise, we're using ATP from the light reactions as a source of energy for the Kelvin cycle. Let's take a look at our diagram. This is the diagram that you should memorize, and we're focusing now on the Kelvin cycle, right, on this half of the diagram. So far, remember, in the light reactions, light energy was used by the photopigments, was absorbed by the photopigments, and used to make NADPH and ATP. That required water. Water is split or oxidized in this in the light reactions. Water donates electrons to NADPH, and a byproduct of that is oxygen. So we make ATP and NADPH. They're going to both be used in the Kelvin cycle. In the Kelvin cycle, carbon dioxide is grabbed, is fixed, and put onto an organic molecule, and then that organic molecule is reduced to make a sugar. Glyceraldehyde 3-phosphate, actually. Okay? ATP is used up in the process. NADPH donates electrons in the process and is recycled to NADP+. All right, now we just have to look at the details. Yay. The Kelvin cycle is divided into 3 main phases. First, you fix carbon dioxide. Second, you reduce and make a sugar. And third, you regenerate. So we need to sort out what that all means. Here's another picture of a chloroplast, and what's happening are the light reactions in the Kelvin cycle in the chloroplast. I do notice that I've got something here, Rubisco. Rubisco's role as a catalyst. Okay, Rubisco is going to be pretty important, and it's actually an enzyme called ribulose bisphosphate carboxylase, or Rubisco for short. We're going to need this Rubisco. Let's look at when we need it in the Kelvin cycle. This is the scariest diagram. Don't worry, we're not going to learn every detail of this diagram. But when we look at a diagram like this, I first look at things that are big and bold to see kind of what's happening. I see 3 phases. I see a circle. So we start with something, we end with that same thing. It looks like we start with ribulose bisphosphate and we do a circle and we end with it. What else do I see? Oh, I see sugar popping out. I see carbon dioxide entering the cycle. I see ATP being used up at different places in the cycle. I see NADPH being oxidized. This must be a redox reaction. It's losing electrons in that part of the cycle. What else do I see? Oh, I see this guy, and apparently I really am highlighting this, Rubisco, and there's a picture of it. It's a big enzyme that is involved in, it looks like, bringing carbon dioxide into the cycle. Rubisco's job is to catalyze the fixation of carbon dioxide into an organic molecule. That's the first step. That's really where we start this whole cycle, and that's called carbon fixation. So we've got this enzyme that's sitting in plants, inside the chloroplasts of plants, that has the ability to grab carbon dioxide and add it or attach it to a carbon-based molecule. And what we have here, we don't even know the name of it. We are not learning that. We're just learning the basics here. So Rubisco is the enzyme that catalyzes the fixing of carbon dioxide into an organic molecule. It's added to that. Actually, what is it added to? It's added to ribulose bisphosphate. And ribulose bisphosphate has, count these little gray circles, that's the number of carbons they have. Ribulose bisphosphate has 1, 2, 3, 4, 5 carbons. It becomes connected to carbon dioxide. And we end up with a 6-carbon intermediate. Yay, Rubisco's job is to grab carbon dioxide, okay? Now, what happens to this intermediate? And also, why are we following 3 carbon dioxides going through this cycle? Why aren't we just following 1? That seems odd to me, but we'll think about that in a minute. So we're following these 3 molecules of ribulose bisphosphate connect with 3 molecules of carbon dioxide to make 3 6-carbon intermediates that immediately break in half somehow, and we get 6 3-carbon molecules with a phosphate on it. We use our ATP up to make a 3-carbon molecule with 2 phosphates on it. This is not a sugar yet. This thing is called 1,3-bisphosphoglycerate. We've finished phase 1. Now we move into phase 2. Now we make the sugar. This is the redox reaction that we see. In this redox reaction right here, NADPH is being oxidized. It donates electrons to 1,3-bisphosphoglycerate, and we make something called glyceraldehyde 3-phosphate. That's a sugar. Yay! Confetti, okay? We have been following how 3 carbon dioxides enter this cycle up at the top here, and we make 6 3-phosphoglycerates. We make 6 1,3-bisphosphoglycerates. We make 6 glyceraldehyde 3-phosphates. Only one of those glyceraldehyde 3-phosphates pops out as a product, a sugar. We made a 3-carbon sugar, 1. What happens to the other 5 glyceraldehyde 3-phosphates? Where do they go? Well, it turns out we need to regenerate ribulose bisphosphate if we want this cycle to go again and again and again. And that takes a bunch of steps. I see lots of arrows there. And it takes up some ATP on top of it. And we need 5 of these sugars, 5 of these glyceraldehyde 3-phosphates to be converted into 3 ribulose bisphosphates. 5 times 3 carbons equals 15. 3 times 5 carbons equals 15. The carbons are conserved. What we're following in this diagram is how 3 carbon dioxides are needed to make 1 3-carbon sugar. That makes sense if carbons are conserved. We need 3 new carbon dioxides because there are 3 carbons in 3 carbon dioxides to make 1 3-carbon sugar popping out. The rest of these materials have to be used to regenerate ribulose bisphosphate. Whew. Now, quick question. Where did we get the ATP and the NADPH from to do the Kelvin cycle? We got it from the light reactions. Remember, light energy was converted into ATP and NADPH in the light reactions. Okay. And for each 3-carbon sugar that is released, how many molecules of carbon dioxide do we need? Well, we just talked about that. For each 3-carbon sugar popping out, we need 3 CO2s. Well, that's a terrible number, 3. You guys are going to have to work harder than me. Oh, I'm having terrible trouble. Write a 3 down there. There we go. How many ATP do we need? Let's count it up. I see 6 over here and 3 over here. We're going to need 9 ATP. Yeah, it's not liking me. 9 ATP for each 3-carbon sugar popping out. And how much NADPH? It looks like 6 right over here, right? Okay, we get our ATP and our NADPH from the light reactions. We get the carbon dioxide from the environment thanks to the enzyme called Rubisco, or ribulose bisphosphate carboxylase. It adds a carbon to ribulose bisphosphate. That's its job, right? This is one mechanism that plants can use to make sugars. Okay, this mechanism happens to be called the C3 mechanism of fixing carbon. There are different plant mechanisms. There's one called the C4 mechanism that works a little differently, and one called CAM that works differently yet. But this is the primary mechanism that most plants use to grab carbon dioxide and fix carbon dioxide and make sugars. This diagram here shows us kind of the numbers involved and how the numbers make sense. So if we take 3 carbon dioxides coming in, we end up with 1 3-carbon sugar going out. And then the rest of the carbons are all recycled. Okay. So the 3 phases again are fixing carbon, reducing, doing a redox reaction to make the sugar, and then regenerating the carbon dioxide acceptor, which was ribulose bisphosphate, that can accept the carbon dioxide from the environment. Wish we could do this, right? It's a pretty cool thing that photosynthesizers can do. Now, you should be able to look at this diagram and fill it out, and use this as your summary for photosynthesis. We've covered the light reactions and the Kelvin cycle, and I think we're in good shape. I will try to post a set of review questions about photosynthesis that should help you as well, and I'll post that on our Blackboard site. We are not covering adaptations in plants, although it's really interesting to look at plant adaptations, especially in their mechanisms of photosynthesis. We are just not covering that this year. And that's it. Thanks.